So we're talking about our biomolecules. We're thinking about their structure, their function, and how they work. We talked about the basic types. So we're thinking about carbon-based molecules, some oxygen, hydrogen, nitrogen. We have our 4 basic kinds. So on Monday, we talked about our carbohydrates. I'm clicking through. And we started talking about our lipids. So we went over what a triglyceride is on Monday. Glycerol plus 3 fatty acids. We can have saturated fatty acids, which have all single bonds in those carbon-hydrogen chains, in those fatty acid chains. Or we can have unsaturated fatty acids, where we'll see at least one double bond in that fatty acid. Our hydrogens will either be on the same side or on the opposite side of that double bond. If they're on the same side, cis. If they're on opposite sides, trans. So our next group of lipids are ketones. So ketones are actually what we get when we start to break down those fatty acids. So they're important to think about when we're thinking about diseases like diabetes or just general low carbohydrate diets where we're breaking down lipids for energy. So ketones are produced by the hydrolysis of triglycerides in adipose tissue. So remember, hydrolysis, hydro is water, lysis is breaking apart. So when we break apart triglycerides by adding water, we're going to start getting ketones. So the process for this is that those fatty acids get released in the blood. They go to the liver, and then at the liver, we produce this molecule called a ketone. So our basic structure here for a ketone, I would like my pointer. No, okay. There we go, pointer. So the basic structure of a ketone, we have a carbon double bonded to oxygen and then 2 side groups. And so here we see a common example of a ketone in the human body, here on the bottom right. Here we see acetoacetic acid. So we see a carbon double bonded to an oxygen, telling us it's a ketone. Here we see one R group, which actually looks the same as that base structure. And here we see our next R group. So when we have a buildup of these ketones, which are this byproduct of breaking down fatty acids, we go into ketosis, which can also lead to ketoacidosis, because as you can see, acetoacetic acid is an acid. So this will cause a change in pH. And we can also sense when we have a large buildup here, one of the side effects of going into ketoacidosis is you get something called acetone breath. So you can actually smell a change. It smells kind of sweet, some people say, or kind of like nail polish remover, other people say. So this is a thing to look out for. If you're working with a diabetic patient, this would be a sign of this state. And that's important to be able to notice because it can be dangerous. Okay, so our third type of lipid that we're going to talk about is a phospholipid. And we'll talk a lot more about phospholipids and really be thinking about them when we start talking about membranes, which we're actually going to leave off until after metabolism. We're going to start talking about membranes when we talk about transport, because the membranes really affect what is able to cross in and out of the cell. So it'll kind of pair well there. It's not exactly how it is on your schedule. Okay. So the special quality of a phospholipid is that it's amphipathic. So when we say a phospholipid is amphipathic, we mean that it has a polar part, so a part that's attracted to water, a part that's hydrophilic, and it also has a nonpolar part, so a part that's repelled by water, a part that's hydrophobic. And this is going to affect the function of a phospholipid. And phospholipids are the basis for our membranes. So it's really important to keep this in mind in your head and to kind of understand what this means when we're thinking about how a phospholipid is going to interact with water and other molecules. So if we're looking at a phospholipid here, we'll see that it has a hydrophilic head. So this head of the phospholipid, to which our tails are going to attach, includes a phosphate group, and it's attracted to water, hydrophilic. These tails are actually made of fatty acids. So these are fatty acid tails. They're hydrophobic. So we talked about the fatty acids when we were talking about triglycerides. We know they're just these chains of carbon and hydrogen. So these fatty acid tails are hydrophobic. So these are going to repel water and be repelled by water. This means that phospholipids automatically orient themselves in kind of cool ways when we just drop them into water or when we find them in a water-filled environment. So they can form a couple of different types of structure. The one we want to focus on is the phospholipid bilayer. So in a phospholipid bilayer, we'll see that the part of the phospholipid that's hydrophilic, that's attracted to water, which is that hydrophilic head, that head with the phosphate group in it, that's going to orient towards the water. It likes water. They play along really well. But we're in a whole environment filled with water, so we're going to have water on the other side too. We don't get just a single layer of phospholipids because these tails are hydrophobic. So if we didn't have the second layer, right? So imagine we just had water right here. This would be a problem because these fatty acid tails, these hydrophobic tails, are repelling water. So they'd keep moving around because they're trying to get away from the water. So we get around this by forming a double layer. So we have a phosphate-based head, so our hydrophilic head on one side. Then we have our hydrophobic tails pointing towards the middle of this bilayer. And they line up with another phospholipid end to end. So we have hydrophobic tails touching other hydrophobic tails. And then this bilayer is protected on both sides by hydrophilic heads, or polar heads on both sides are touching water. So in this way, those fatty acid tails, those hydrophobic tails, are nowhere near the water because they're just touching each other. And these polar heads, these hydrophilic heads, are touching the water on both sides. So when we see a membrane, we want to keep in mind that a cell has water both inside it, right? We have intracellular fluid, and it has water outside it. We have extracellular fluid. We talked about the fluids. So we need these hydrophilic heads on both sides of that membrane. So if you're thinking about phospholipids in your head and you're thinking, wait, but why don't they just form a sphere? Why do we need a double layer? It's true, they can just form a sphere, but then there's nothing on the inside. So this isn't how a cell forms. But we do have another structure that phospholipids sometimes form called a micelle. So some of you may have seen like micellar water at the pharmacy for washing your face. This is what a micelle is. So this is a phospholipid or some other amphipathic structure where the nonpolar part is on the inside, just all clumped together, and the polar heads are on the outside. But this is going to be small. It's not big enough for us to have all our different organelles on the inside. And also we can't get water into the center of this because we don't have a second layer. So that's why we want to think about this phospholipid bilayer. Okay. So surfactant is an example of where we would see phospholipids aside from our phospholipid bilayers of all our cells. So surfactant is something that the lungs produce to prevent collapse. It actually lets the membranes in our lungs. And when I say membranes here, I'm not talking about just a single cell membrane. I'm talking about like the surface layer of that tissue, that outside epithelial layer. It allows them to slide against each other. So that's another place we would see these lipids. Okay. Next type of lipids, eicosanoids. So eicosanoids are pretty similar to our triglycerides. So the way in which they're similar is that we see these carbon-hydrogen kind of tails. They're basically modified fatty acids. So they're kind of like a fatty acid almost folded in half, right? So they're a type of lipid that we see in intracellular communication. And some examples that we'll see later in this course when we're talking about specific systems are prostaglandins, which are partially responsible for uterine contractions. And we'll also talk about thromboxanes, which are important when we start to talk about clotting and the circulatory system and how it repairs itself. So just keep that in the back of your mind for now. But we will see examples of eicosanoids much later in this course. It'll take us a while to get to those. Those are really going to come back for us in A&P 2. Okay. Important group right now, steroids. So steroids are derived from cholesterol. So they all have this similar ring structure. So we'll see these sort of 4 rings pressed against each other based from our cholesterol molecule. And this will come back pretty soon because we're going to be talking about our cell membranes. And cholesterol, we'll see, is one of the components of our cell membranes. But steroids actually have a lot of jobs in the body. And an important one, aside from being a constituent part of our plasma membrane, is that steroids act as hormones. So some hormones are proteins and some are lipids. That's not something you need to write down for right now. It's just a thing to know in your mind if you're thinking about it for now. But our lipid hormones are going to have this structure. They're going to be based on steroids. So some examples of steroids we'll see, aside from cholesterol in our membranes, are estrogen and testosterone, our sex hormones, also aldosterone. So we'll see steroids coming back in our future units as well. Okay. So our next group are proteins. So proteins, we're really going to talk about a lot in this course. So proteins are polymers. They're big, long chains of amino acids. And they have a lot of functional jobs in the cell and in the body. So when we're looking at the structure of a protein, the structure is going to have a number of different levels because one of the things that's really important about proteins ultimately is their shape. So when we're starting out building a protein, building blocks of proteins are something called amino acids. There are 20 different kinds of amino acids. We link them all up in a line. You do not need to memorize the 20 different kinds of amino acids for the purposes of this course. So we're going to take a look at what those amino acids look like as we start to build them into a chain. So here we see 2 amino acids bonding together. We have a covalent bond between the carboxyl group of one amino acid and the amino group of the next amino acid. So if we're trying to recognize a protein, a thing we can look for is this nitrogen. Okay, so we didn't really see nitrogen when we were talking about carbohydrates, or really when we were talking about lipids. That wasn't an important component to look for in those 2 types of macromolecules. But it was one of the atoms that we listed, one of the elements that we listed when we were talking about biomolecules. That's because nitrogen is a super important component of proteins. Okay, so we link together amino acids into a polypeptide chain. So here, peptide really means the same thing, polypeptide. Peptide is just a general term, number of different lengths. So peptide will be generally about 2 to 50 amino acids. We start to use the word protein when we're talking about a really big chain that starts to fold in all sorts of different ways. So a protein will be more than 50 amino acids. Don't worry about memorizing those numbers. I'm just clarifying here why you'll sometimes see one term and why you'll see the next term. It's really telling us how big that molecule is. And we can see that this is also a condensation reaction when we link together these 2 amino acids into our peptide. So as we start to talk about our proteins, so our big molecules with shape and therefore a crazy number of different possible functions, we build that shape in different steps. So the first level of structure for a protein is the primary structure. And that's really what we were just talking about as we were talking about linking together amino acids. So the primary structure of our protein is just, we got one amino acid linked to amino acid 2, 3, 4, and so on. So our primary structure is all bonded together by peptide bonds. That's the name of the covalent bond between our carboxyl group and our amino group, which is why you see the nitrogen right here next to each of our peptide bonds, because that's from our amino group. Primary followed by secondary, right? We're just counting 1, 2, 3, 4, basically. So the secondary structure of a protein comes in one of two shapes. So once we have this long chain of amino acids, it starts to curl up. So some parts of that primary structure will form helices. So a helix is just a spiral, right? And when we're talking about proteins, we call it an alpha helix, basically because we're naming these shapes like A, B, and really we only get to B for our proteins. So parts of this secondary structure will coil into alpha helices, and other parts of this structure will fold into sheets. So sometimes we'll see a curl, and sometimes we'll see the peptide chain double up on itself and form what we call a beta pleated sheet. So that's our secondary structure of the protein. That's the first step as we start to crumple it up. Sorry, that's the second step. Yeah, no, that's the first step as we start to crumple it up. Our tertiary structure continues basically the crumpling up. So we can see here that we have alpha helices and beta pleated sheets. So those were part of our secondary structure. Now we can see that we've started to basically crumple up those alpha helices and beta pleated sheets into like a messy kind of ball. So that's our tertiary structure as we continue to get more varied shapes, basically, as we start to make this structure more three-dimensional. So when you're thinking about the tertiary structure of a protein, you'll see components that look like these alpha helices, you'll see components that look like these beta pleated sheets, and you'll also see some sort of connector regions that don't look exactly like either of those shapes. Now finally, we'll have a quaternary structure for our proteins. And the quaternary structure is basically that we have multiple polypeptides working together. So this is a picture of hemoglobin. So here we see in red the different pieces of hemoglobin, and it actually has 4 pieces. So that's why we can see that 2 of these shapes are dark red and 2 are light red. That's trying to show you that we have kind of 4 components working together. So we would have had one polypeptide chain turning into alpha helices, beta pleated sheets, then getting crumpled up into a tertiary structure. And then we would have had a second chain doing the same thing, third one, fourth one, and then they all join together to form this quaternary structure with different subunits, which works as our fully functional hemoglobin molecule. Okay. Finally, our last type of biomolecule, our last type of macromolecule is the nucleic acid. So when we're talking about nucleic acids in biology, in AP, we're talking about 2 specific molecules. Basically, we're talking about DNA and we're talking about RNA. They both have similar structures. So if you break down any nucleic acid, it's going to be able to be broken down into these structures called nucleotides. So a nucleotide is made up of some specific groups. So we have a phosphate group, we have a 5-carbon ring. So here's sugar in the center of our nucleic acid, but a nucleic acid is not a carbohydrate because it has other parts. So we have our 5-carbon ring, and then we also have a base. So our base, we may also call nitrogenous bases. They'll contain nitrogen, and they'll come in different shapes. So some of them will have 1 ring, some of them will have 2 rings. So we can separate them into our pyrimidines or our purines. And we have 5 total, lumping together the nitrogenous bases we'll see in DNA and the ones we'll see in RNA. So for our pyrimidines, we have cytosine, thymine in DNA, and uracil in RNA. For our purines, we have adenine and guanine. So aside from the fact that DNA has thymine and RNA has uracil, one of the other important difference between DNA and RNA when we're talking about their nucleotides rather than their bigger structure is what that 5-carbon sugar, that 5-carbon carbohydrate ring looks like. So in DNA, which is deoxyribonucleic acid, we have deoxyribose. And in RNA, we have just plain old ribose. So RNA stands for ribonucleic acid. And the difference between these two things is really just these 2 functional groups we can see here down at the bottom of the ring. So we can see on ribose, we have 2 hydroxyl groups. So we see OH here, we see OH there. So we got 2 of them. Deoxyribose, our DNA nucleotide, doesn't have an oxygen here, right? So we have 1 hydroxyl group, but then our next group is just a hydrogen. So we have 1 less oxygen, which is why it's deoxy. We got rid of an oxygen. As we move into metabolism, we will see another type of nucleotide that's going to be really important. So these aren't DNA or RNA. There's something we call energy transferring nucleotides. And we'll see a couple important kinds. The most important one is ATP. So adenosine triphosphate, which is our form of cellular energy. So we'll see that in all types of cell functions because it's basically how we get cells to do stuff. And when we're talking about metabolism, we're talking about, well, how do we make that energy? How do we make that ATP to run our cell functions? And we'll look at a couple other types of energy transferring nucleotides that help us make ATP. So in addition to ATP, we won't really see AMP often, so that's adenosine monophosphate, but that's not really going to feature in our story much. But we will see a lot of ADP, adenosine diphosphate, okay? So that's going to be one of our precursors to ATP, because AMP, ADP, and ATP are all grouped together because we're just listing how many phosphate groups there are attached to this molecule. So here at the bottom, we see a picture of ATP, adenosine triphosphate. You can see the P's here, right? So P, P, P. So we got 3 phosphate groups. If we rip one of those off and we leave ourselves with just 2 phosphate groups, it becomes adenosine diphosphate, di for 2. And if we were to rip off even more phosphates and just leave ourselves with 1 phosphate group, that's when we'd have adenosine monophosphate, AMP. And we can do this in the reverse, and we will do this in the reverse as we're talking about metabolism. We'll start with ADP, adenosine diphosphate, with just 2 phosphate groups, and then we'll tack on an additional phosphate group and give ourselves ATP, adenosine triphosphate. So that's really what our goal for the whole process of metabolism is. So our helper molecules for this that are also nucleotides are NAD and FAD. So there aren't pictures of them up here. Honestly, you won't really have to see pictures of them, but they look relatively similar. So we also have nicotinamide adenine dinucleotide. So it's just another type of nucleotide, and it'll work the same way, basically. We'll be able to remove parts of it and add parts of it. And we'll also have flavin adenine dinucleotide. We'll sometimes remove parts of it. We'll sometimes add parts of it. And we'll talk about exactly what we're removing and adding when we start talking about metabolism. that those are going to work together to form ATP. Okay. Cyclic nucleotides do also exist. The important ones that we want to think about in this course are messenger molecules. So when we start talking about cellular messaging, we'll be talking a lot about cyclic AMP, so cAMP. So these are messenger molecules basically inside the cell that allow us to amplify signals and to get the cellular function inside the cell, kind of upregulating different processes. So if we were to look for what these cyclic nucleotides look like, they basically form a ring with the phosphate group. So we can see here that we have a phosphate group, but it attaches back down over here to our sugar. So that's why it's cyclic. That's why we have this C here at the front. Okay. We're going to start enzymes in a second, but questions about this review of biomolecules before we move forward? I know I was talking for a long time there. All right, great. Let's talk about enzymes. So we want to talk about the role of enzymes in chemical reactions. So enzymes are a specialized type of protein that are going to allow us to basically speed up things going on in our cell. So speed up chemical reactions in our cell, because we're biologists, we want the body to work, we want our cells to do things, we don't want to have to wait around for a chemical reaction to just happen spontaneously. We want to be able to control it and to control its rate. Okay. So we have some general ways that enzymes work. So they're proteins speeding up reactions. So in scientific terms, we'll say that they're proteins that are catalysts for reactions in biological systems. So catalyst, hopefully you remember from chemistry. I saw from the poll question, you're all chemistry whizzes, which is great. So a catalyst is something that increases the rate of a chemical reaction by decreasing the activation energy for that reaction. So the way this works, here we have at the bottom, we have an example equation for just a general chemical reaction. We didn't put on any specific molecules, right? But the basic parts of the chemical reaction that should be familiar, right? We have our substrate or our reactant, whatever we're starting with. So our substrate. Then we have some arrows, and eventually we have our product here on the right side. But because we're talking about the function of enzymes, because we're talking about reactions that are catalyzed, we see that we add in the enzyme at the beginning to the substrate or our reactant, same thing. We form an enzyme substrate complex. So for a little while, our enzyme and our substrate are stuck together. That's how the enzyme is going to be working. And then we have our product plus the enzyme. So eventually something breaks off from that enzyme, the enzyme is released, and that's what gives us our product. And we'll see that these are double arrows, so they go both ways. So from this enzyme substrate bound state, we can get our enzyme and substrate back again, or we can get the product and enzyme. What I'm saying is that this equation is reversible. It can work in both ways. And which direction it works is going to be partially dependent on the concentration of that substrate and the concentration of the product. So by the law of mass action, so the law of something called mass action, basically says it's going to go in the direction where we have a lot of something, and then we're going to make whatever we have less of. So if we have a lot of substrate, we're going to convert it to the right here. We're going to convert it to product. But if we had a ton of this product and not very much substrate, we might have this reaction go in the opposite direction. So to the left, we might have this product actually flip back and turn into the substrate. So that's why we have those bidirectional arrows there. So we're going to end with a drawing. So we'll pick up with enzymes again on Friday. But before we go, I want us to draw together an activation energy barrier. So I basically want us to do a sample graph of a chemical reaction. So I want us to draw energy on the y-axis versus our reaction coordinate on the x-axis, which we can just think of as time. So if I flip back for a second, we're going to think about this equation moving to the right here, even though we know it could also move to the left. And we want to plot the energy that's contained in each of our components, so in the reactants and in the products. So I'm going to let you guys start drawing, but I'm also going to pull up my camera and then draw with you in a second. So we're drawing activation energy in a reaction. So over here on our y-axis, what we're concerned with is the energy. Over here on our x-axis, we're talking about the reaction coordinate. Which we can basically think of as time. Reaction coordinate is more specific and slightly more correct, but I find it helpful to kind of think about it just as the progression of this reaction over time. So pretty basically, what we should be drawing, we're going to start with our reactants or our substrate at a certain level of energy. So here we have our reactant. I specified in that list, we're talking about an exothermic reaction, so something that's going to release energy, so something that has lower energy. So at the end of this process, we're going to end up with a product that has less energy. And the way our typical reaction works is that in order to have this reaction progress, we can't draw just a straight line from the reactant to the product. We have to reach a certain activation energy, so I'll have a little peak in the middle here, before the reaction can proceed and form our product. And the whole point of enzymes, so this is our activation energy. The whole point of enzymes is that we want to lessen the amount of energy we have to accumulate in order to have this reaction proceed. So the way we speed up the reaction is by lowering this activation energy barrier. It's what a catalyst does. And when we're talking about biology, our catalyst is an enzyme. Okay. And I know we're over time, so we're gonna stop here. I'm going to hang on the line for a bit for anybody who has questions, whether it's about your lab schedule, about this material, or about anything else. But you are free to leave and live your lives now. Hope you all stay healthy for the rest of the week. I know there have been some kind of scary emails going out. I hope you're all doing okay.